Let's start this recording. Ooh, whoa, sorry. I got scared by someone entering loudly. All right, so today's virtual session is going to be pretty short, just because we're going to be focusing on doing our discussion-based assessment, or really just reviewing and making sure that you understand all of the material from this module. And I'm going to talk a little bit about what's coming up next. I'm going to be publishing a lot of material later this week and this weekend for the next module. So it's going to be pretty intense, and I would argue maybe the hardest module is coming up next. So today's objectives are really to master vocab and understanding from this module on macromolecules. I say master because, you know, I mean, really, you want to be ready. So we are having a module exam at the end of this module. The next two modules after this will have final projects instead of exams. So this is the last exam. The reason why I chose to do an exam for this module is because it's a lot of vocab and it's a lot of basic stuff. Do you understand this? Do you understand that? If you know what all the vocab means and you understand the basics, it's going to be a lot easier for you to then critically think. And I started pushing some of you guys on critical thinking already with some of the questions about DNA, right? This class is not going to be a class where I tell you the answer to everything, right? That's not fun for me or you. So make sure, right, that you are putting on your thinking cap here, because some of these questions will require you to take knowledge and apply, right, or think about why something might be the way that it is. So the goal for today is that we're going to talk about 5 of these questions at the very least. We may talk about more if you'd like. What you're going to do to get credit is you're going to be turning in at least 3 questions to get credit for this virtual session, right, as your review sheet. I'm also going to be keeping track of who talks, right? I didn't make a rubric for this one, but I want to push everyone to respond to at least one question or ask a question or say something, right? So that we all are participating. And if you're one of those people, Lane, who loves to talk in the virtual sessions, right, then you're going to work on the opposite, right? Trying not to be the person who's answering every single thing, right? Take a step back, take a breath. Analyze the situation a little bit, right? Because we want everyone to kind of be able to talk here. And I know it's hard because I'm just like you, Lane. I love to answer every question, right? So for me, it's hard to be quiet. So whichever one of those things is hard for you, focus on trying to do the opposite today. Of course, still participating at least once. So I've made a list of all these questions, and these are the questions that I used to create the exam. The exam has some short answer questions, some multiple choice questions, some matching questions, right? Again, a lot of this is just building that foundation of vocab, because the next unit we are going to talk about processes that all of these molecules are involved in. And that's where it's going to start to get a little bit deeper. So you guys are going to pick the questions we go over. So right now, as part of your warm up, right, I'm going to assume some people have never seen this screen before because they didn't look at this yet. If you have, right, I want you to think right now, by looking at these questions, what is the first question that you would like for us to go over as a group, either because you don't know the answer, or you want to make sure that you're correct, or you just want a little bit more explanation. So right now, taking a few moments, read through the questions, pick your number 1 question that you want to discuss as a group. Hi, Joelle. Okay, great. Colin already typed one in there. So we're going to start with Colin's question. We'll give everybody else, though, a little bit of time here before I start talking about it to populate with their own question that they would like to go over. Oh, good. I love people just type in the numbers. 8, 4. Okay. Colin's is 6. Okay. 4, we got another vote for 4. Bonnie says 14. Oh yeah, okay. 4 and 7, we also got 7. So I've got 5 here, right, to start. Question 8, question 4, question 6, question 14, and question 7. If there are other questions that you want to go over, we'll go over them too, right? But those are the 5 that we'll definitely start with. All right, so let's start with question 8. Because that's the first one that I wrote down in terms of numbers, excuse me. What is DNA made of and how is it arranged? So this is a deceptively easy question, right? A lot of people on their DNA questions wrote, it's a double helix, it's a nucleic acid, it has nucleotides, right? We want to be as specific as we can. So let's see, what do you guys think? What are some details of DNA structure that should definitely be mentioned here? Okay, the double helix is important, right? That is one component of the structure. The base pairs, Georgia says, which I think is what Lane was trying to get at here, it has 4 chemicals in the middle. What are those called? So they're called technically nitrogenous bases, right? Sometimes you'll just see them referred to as bases. And Georgia just did a great job of listing them as adenine, thymine, cytosine, and guanine. Now, do you have to remember each name? Not necessarily, right? The letters that represent them are going to be much more important for our purposes here, because you'll always have your notes to look at. If you want to remember what C stands for, you can look that it stands for cytosine, right? So make sure you understand which letter represents which one. It's just based on the first letter, right? So adenine is abbreviated by an A, thymine by a T, cytosine by a C, guanine by a G. These are the nitrogenous bases. Those are in the middle. Okay, so that's an important part of the structure. What else is in the structure? What actually makes up the backbone of DNA? In fact, I'm going to go to a blank tab so that you guys can see what I wrote easier here. Protein is potentially a good guess, but not quite. Sugar phosphate. Thank you to Colin and Bonnie and Georgia, who all said that, right? There's a sugar phosphate. It's often referred to as a backbone. Phosphate, backbone. Which really, if we look at any picture of DNA, nice little Google Images search here, right? Most of these are not like a super detailed one. I believe I used this one in the lesson, actually. We see the difference between DNA and RNA a little bit here, but we see the sugar phosphate backbone is just one color here. It's just gray, right? A lot of pictures simplify it, but it is composed of those 2 components, a sugar and a phosphate, right? That's going to become important because when we differentiate between DNA and RNA, the sugar is going to be different. The sugar does matter, right? For DNA, the sugar is deoxyribose, right? It's a deoxygenated ribose sugar. RNA just has a ribose sugar. They are different, right? That is one of the main structural differences. Okay, what else is in the structure though? There's another word here that is a super important word that no one has mentioned yet. It starts with an N. Nucleotide. Nucleic acid is a good guess. Nucleotide refers to sugar, phosphate, nitrogenous base. So it's those 3 things together makes one nucleotide. Nucleotides are the building blocks of DNA, right? And other nucleic acids. So a nucleotide is not just the base, it's the base and part of the backbone. So again, if we look up what a nucleotide looks like, nucleotide. this is a pretty important thing for something we're going to talk about later, right? But this is a good example of a nucleotide. It has a phosphate group, it has a sugar, and it has a nitrogenous base. Right now, it doesn't matter which base. It could have A, T, C, or G. It could even have U if it's RNA. But it's any base. So these three pieces together make one nucleotide, and nucleotides make up DNA. Okay, so let's look back at our question to make sure that we have answered it here. I hate how in Zoom there's like so many little bar things that I can't see. Okay, question 8, what is DNA made of and how is it arranged? Okay, so if we now know all of these things, how could we describe how DNA is arranged using all of these words? I'm going to challenge you right now to be able to do that. I'm busy writing down people who have contributed to the conversation right now. And I have to say, seems like we've got quite a few contributors today. What was the question? Hey, no, that's a fine thing to say. So what is DNA made of and how is it arranged? What can we say now that we've sort of talked about all of these components? Okay, Colin's suggesting a great answer to this. It's arranged with the sugar phosphate as its structure with the 4 base pairs as the central components. Good, right? I gave another example here. DNA is composed of nucleotides. Each nucleotide has a sugar, phosphate, and 1 nitrogenous base. I put based accidentally. The bases are, and then you could mention them, right? Cytosine, guanine, adenine, thymine. Really, anything that you say is fine as long as you include the word nucleotide and you somewhat talk about the structure, right? Is it a double helix? How are they arranged, right? Colin gave us great descriptive words that the bases are in the center, right? They're the central component. As long as you have some kind of descriptor of where things are located. Let's see, Bonnie's got the bases in the middle are held by weak hydrogen bonds, very important. The base pairs have certain ways that they're going together. Good. Adenine, thymine go together. Cytosine, guanine go together. They always pair this way. It's made of helixes and nucleic acids and different phosphates and strands of atoms and stuff. It contributes to create a strand of DNA and the bases combine with the DNA strand to make it whole. Okay. DNA is made up of nucleotides. That's like the simplest but 100% correct answer. It's made of molecules which are, or it is a nucleotide. Good. Good, Colin edited and added to his a little bit here to include the weak hydrogen bonds. So that is a question that no one asked us to go over, but I kind of want to mention that really quick. I emphasized that in the lesson, right? And I kind of asked you guys why DNA is structured the way it's structured, which some people were kind of like, well, because it is. Well, yeah, right? I mean, we know that now, but we didn't used to know what DNA even looked like or did because we couldn't see it, right? It's really small. We had no idea what was going on. Why would the most important molecule, arguably, inside your body, right? It contains the code to make you, have weak bonds in the middle of it. Why would it be like that? You would think that DNA would be so strong and impossible to break, like a diamond, right? Because you wouldn't want it to get destroyed. So how come these hydrogen bonds are what hold DNA together? Bonnie says it in the simplest way possible, replicate. Yeah, right? DNA needs to be accessed. If it was a molecule like a diamond that you couldn't get in, then you wouldn't be reproducing, right? You wouldn't be making more cells. You wouldn't be able to do anything because it wouldn't be readable. Exactly, Colin, right? So those hydrogen bonds are important because it allows DNA to be broken. Now, at the same time, that is exposing DNA to the elements, right? Which is part of why complicated cells have a nucleus, to protect the DNA more. Also, honestly, your DNA is getting messed with at all moments of your day, right? Right now, your DNA has probably already had to repair itself thousands of times. Anytime that you go in the sun, which I know it's not really sunny today, so kind of not a good example, every time the sun interacts with your cells, it breaks your DNA and causes issues, and then your body fixes it, right? So your body is constantly fixing your DNA, constantly. Which, guess what? If you're constantly fixing something, at some point there's going to be a problem, right? Which is what's going to lead us into some of these later things. We're going to learn about these processes, and then we're going to learn about the problems, right? And what happens when it doesn't go how it's supposed to go. Right, so anytime that you are exposed to even your computer, right, or radiation from your phone even, that can impact your DNA. So there's a ton of things, right? And that's a little bit, getting ahead of ourselves, so I won't go on about it much more. But those hydrogen bonds are important because we need to be able to access DNA. It needs to be accessible, even though we do want to protect it. Okay, that's question 8 pretty thoroughly. Question 4, explain the difference between a eukaryotic and a prokaryotic cell. Now, I did not necessarily have this in the lesson. I don't believe I even put this on the quiz, but this is something that's good to know that will come up later and that you might see in writings about science, really, right? Because you might end up seeing something say in eukaryotic cells, blah, 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 blah. So we should know what that is. So does anybody have an idea of what a eukaryotic cell is? Either you looked this up maybe when you saw it or you learned it in a previous science class. What is the difference between eukaryotic and prokaryotic? And we're even going to do a little bit of Greek analysis. These are Greek words, what these words actually mean. So if you do know what they mean, go ahead and type in the chat. I'm going to start talking about the Greek. So what we know from Greek is that karyo, it means kernel. So it's talking about a kernel, which in this case is referencing the nucleus. So karyo equals kernel, right? Like a kernel of corn, essentially, like a tiny little piece, which in this case is what the Greeks were referring to as the nucleus. Eu as a prefix means true. So eukaryotic literally means true nucleus. These are cells that have a nucleus. So Georgia is correct, right? Eukaryotic cells would include plant and animal cells, right? Because they have a true nucleus. They have membrane-bound organelles, is something else that you might have heard. Eukaryotic cells are more complicated, right? They have chloroplasts and mitochondria and vacuoles and all these other components of a cell that prokaryotic cells don't have. Prokaryotic, right, again, karyo means kernel, pro meaning, actually don't even know what the prefix pro means. I think it means no, must. But prokaryotic cells don't have a true nucleus, right? So they are going to be, in this case, bacteria, pretty much, right? Bacteria are basically the only cells that are prokaryotic. Every other type of cell you can think of is eukaryotic. Fungi, protist, plant, animal, all have eukaryotic cells, right? Meaning they all have a nucleus and they all have membrane-bound organelles. Prokaryotic cells are just bacteria. I think pro means like original or something like that. It's like first. Bacteria were the first cells, right, arguably. So bacteria still have stuff in them, right? They have ribosomes, they have DNA, they have a few other like enzymes that can function like organelles can in other cells. So when you see the difference between those two cell types, right, just think about really the difference between bacteria and everybody else is really what we're talking about. We will see this Greek karyo word come up again, right? We'll talk about karyotypes, which are a way that you can look at chromosomes, again, related to the nucleus. Okay, so that's the difference between prokaryotic and eukaryotic. Eukaryotic cells have a true nucleus. You can even remember this by thinking you as in you, right? So what kind of cells do you have? Well, you have eukaryotic cells because your cells have a nucleus. So Other questions that we're going over. Question 6. What process does a ribosome do? I guess that should be which, huh? You have to change that. And which macromolecules are involved? All right, so does anyone have any ideas for any part of this question? Do we know what a ribosome does, first of all? Oops. I'm going to go ahead and say, Lane, if you know this one, I know you've been working hard over there to not say the answer to everything, right? But if you know this one, go ahead. What does a ribosome do? This could be solved by a simple Google. Creates proteins for the cell. Thank you, Briley. So, creates proteins, right? Yep, that's what they do, right? Ribosomes make proteins. Ribosomes are in all cells. Bacteria, plant, animal, fungi, protist, anything. It has a ribosome. What do they look like? Ironically enough, they look like hamburgers, right? They sort of look like this. My beautiful art skills. Okay, that's fine, Lane. So ribosomes look like little hamburgers, right? They have this space in the middle where we will learn later where the mRNA actually slides right in. And then it really kind of looks like a hamburger with lettuce coming out of it, essentially, although it's all molecular stuff. So ribosomes do make, you are allowed to, I'm just saying don't answer every question in the chat, but in your mind answer them. So ribosomes create proteins, that's the first thing, right? If you never remember a single organelle for the rest of your life, ribosomes are probably the most important ones to remember, right? Arguably, mitochondria are important, right? Chloroplasts are important. There's other ones that are important, but ribosomes are kind of those organelles that people forget about, but yet they do everything that you're supposed to do. They make a million proteins so that you can function. So which macromolecules are going to be involved in the process of making proteins? Think about what makes up a ribosome. If you've done the DNA, RNA lesson, right, you should already have learned about which macromolecule actually makes up the ribosome. Cell membranes is a great question, or with a question mark. Is a great guess, no. Nucleic acids, yes. Specifically, which nucleic acids make up a ribosome? RNA, good. Which kind of RNA? rRNA is the specific type of RNA that creates the ribosome. It's kind of easy to remember because it just has an R in front of it, right? Ribosomal RNA, rRNA. Sounds weird to say, right? rRNA. But RNA makes up the ribosome. That's why it can be in every cell. It doesn't have a membrane, right? Some organelles do. In fact, they function because they have membranes. The ribosome doesn't need that, right? The ribosome is composed of RNA, which is a nucleic acid, which a lot of people got that component. Great. So we know that rRNA is involved in actually creating the ribosome. Yeah, it kind of looks like a typo when you first see it, right? You're like, rRNA? Did someone just type R twice? No, like it's supposed to be called that. Now, what other macromolecules are involved? Well, I'm going to list some out for you guys, and you're going to tell me how they're involved. And Isaiah even beat me to one of mine. mRNA is 1,000% involved. So how is mRNA involved? What does it do? Based on just the very loose understanding that we have so far, what does mRNA do? It's a messenger, right? mRNA's entire purpose is to take the code from DNA, go to the rib, also known as the ribosome, and give the message, right? We're not going to send DNA out into the cell. Terrible plan. We're going to send a messenger, and if it gets destroyed, oh well, we'll send another one. Right? So mRNA takes the message to the ribosome. The ribosome is made up of RNA as well. And then how is tRNA and or amino acids involved? Carries a message from DNA to ribosome, good. Yeah, the body is better at communication than you are. Yeah, right? It's actually figured this stuff out by trial and error, most likely. So tRNA transports the protein close, right? tRNA brings the amino acids over. So tRNA is kind of like the party planner who's running around grabbing all the stuff that you need to make your party, which in this case is the protein party, right? So tRNA goes, oh, we need that amino acid. Here it is. Oh, we need that one. Okay. Oh, here's the next one. Here's the next one, right? And it assembles the protein using amino acids. It sounds like a lame party, but it's actually awesome because then it allows you to exist. So arguably it's cool. We will study that process in a lot more detail, right? But that's a pretty glossed over version. RNA, all the RNAs are involved in this. And amino acids are also involved in this, right? Which we know are the building blocks for proteins. So we have protein and nucleic acid interacting together in the ribosome. And again, proteins are really what create traits, right? That's what allows you to look how you look, act how you act, do what you do. So it's very important. And we're talking about on the microscopic level here. Like imagine how many proteins you have to make to create one strand of hair. It's a lot, right? A strand of hair is so thin, but a protein is so tiny, you got to make millions of them. So ribosomes are just popping proteins out all day. I like that you guys are the lame party of your life, a sad party than a lame one. Well, hey, it's a party regardless. Okay, so the best answer to that question in summary, right? What process or which process does a ribosome do? Well, it makes proteins. And which macromolecules are involved? Amino acids, which make proteins, and the 3 types of RNA are involved, right? It sounds inefficient. It's not. It happens incredibly quickly, right? And all cells do it, which arguably means that it is the most efficient way for the cell to function. Sounds like it would take longer. Yeah, I think, honestly, what's hard about it is the explanation sounds really complicated, but the cell is just continuously doing it really quickly, right? So imagine like pulling on a string and proteins were just getting produced. That's kind of what happens. Couldn't a genetic engineer create a better process? Possibly. However, right, usually genetic engineering is about manipulating the cell's own processes to do something that we want. So what genetic engineers can do is they can make the cell create certain proteins. Do they have a better way to create proteins? You could build the protein yourself, but it's going to take you even longer to assemble all the amino acids in the correct order. Plus, proteins have to get folded, which is like a insane mom who cares about the way you fold the shirt sort of level of folding. So that's hard for scientists to mimic, is the folding is really complicated because proteins have like 3-dimensional shape to them. That's pretty hard to create without a ribosome, actually, and the folding portion too. But genetic engineering is more about making cells produce things that they don't usually produce because you want them to produce it. And we will talk about that when we talk about genetics, right? But that's a great thought, right? Couldn't we create a better way to do it? I mean, you could try, but it's pretty hard, especially using the machinery that's in the cell. Once we're just RNA floating in a chemical dense and very hot water on an exploding rock with the sun killing everything out of the water. Yeah, right? So all of these processes all started out being for really small organisms like a bacteria, where they just have to make a few proteins, right? Then multicellular organisms appear, right? Suddenly cells have to work together to do stuff. So it does get intensely more complicated as you get bigger in terms of organisms, right? But they all do the same process, which is cool. So that is question 6, a great one to go over because it includes a lot of vocab. Question 14, what happened in the liver enzyme lab and why? Who was in the liver enzyme lab that could explain briefly sort of what we saw or experienced in that lab? So again, in case you were like, wait, did she ask a question because she got really quiet? Yes, I asked a question. What happened in the liver enzyme lab? And why is that? Why did we see that? We looked at bubbly chicken livers when we poured hydrogen sauce or hydrogen peroxide on them, and they were at different temperatures, and we compared them. Mm-hmm. We tested the amount of oxygen released when enzymes were mixed with hydrogen peroxide. Good. Lane sent a link, I'm guessing, to this experiment. So, to something else. Okay, well, keep it relevant. Yeah, so basically, really, the main things I want you to get out of that lab are, 1, liver contains enzymes, right? Liver is an organ. All of your organs have enzymes. Basically, everything in you has enzymes, but organs are a very densely packed enzyme area. And making the liver at different temperatures impacted its ability to work, right? If it was too hot, like it was boiled, it didn't work. It was killed, essentially, right? If it was too cold, it didn't work as well because it was cold. Body temperature and room temperature were the most successful because the temperature wasn't too extreme and the enzymes could function. So really, it's about looking at the temperature and the effect of temperature on enzymes. If you see that question on the test, you're going to want to use the word denatured, right? You're going to want to say, at extreme temperatures, enzymes get denatured, right? When it's too hot, the enzyme can't work. When it's too cold, the enzyme can't work. When it's just right, the enzyme can work, right? Goldilocks kind of taught you a little bit here about enzymes, right? They need it just right. But seriously, they do, right? Other things that can impact enzymes would be pH and salt. We will talk more about that when we talk about homeostasis later in the year. But basically, the main concept right now is temperature affects enzymes. And that's what we saw. And then the why is because they get denatured. Okay, well, I'm glad you're back. All right, last question that I see on here is 7, which guess what? Similar to question 6. Again, a preview. Right now, we're at basic level of understanding. Later, we will go into excruciating detail of what happens in the chloroplast. But which process happens in a chloroplast? I'll give you a hint. It starts with a photo. Photosynthesis, right? Most of us are like, oh yeah, that thing, right? That's what happens in a chloroplast. Cool. Well, which macromolecules are involved? Ooh, this one's a little harder. It's not quite vitamin D to sugar, but that's a good guess. So Lane does point out one macromolecule that's involved here, sugar. Because what are we doing in photosynthesis? What does photosynthesis mean? It means using sunlight to create sugar. Which macromolecules are involved? In this case, we don't have as much detail yet, right? We didn't talk about the specific molecules as much as I did for ribosomes, right, with the RNAs. But in this case, amino acids, yes, but in the form of protein, right? We have a lot of proteins working here. We have a lot of molecules that we haven't talked about yet, so I won't even mention them, but we will talk about them. So really the 2 macromolecules that I want you to know that are involved in this process are proteins and sugar. Sugar is created, right, and proteins are used mostly in the form of enzymes, right? So you might want to write down enzyme as something that is used, because the enzymes as a protein, right, that is an enzyme, it will help create the sugar inside a plant. So we're going to talk about both of these processes, photosynthesis and cellular respiration, which are opposites, right? And we're going to talk about the similarities and the differences and the other molecules that are involved. We're going to go into more detail on that in the future, but right now you should just know that sugar is created and proteins are involved. Carbon dioxide and water are involved. Again, though, not macromolecules, but are molecules that are involved, right? And then eventually, yes, we're going to be going into ATP. You could put ATP as a molecule involved. ATP is technically a nucleic acid, I believe, because it has an adenine triphosphate. Well, it's not made of nucleotides. I guess it's just a molecule. It's not really a macromolecule. It's most closely related to a nucleotide, but it's not quite a nucleotide. But essentially, ATP is something that's involved. So you could list that. But really the two major ones, sugar, proteins. Okay. Does anybody else want to go over a question that's listed on here? Because we're already about 37 minutes in, if you can believe it, just from 5 questions. Most of these shouldn't be too hard for you to answer, right? And of course, if you answer one and you're not sure, email me, right? Say, hey, I don't understand this one, or I put this, is that true? Most importantly, I'm looking for your words, right? I love, for example, when I read Colin's answers because I know that it's Colin's words. No one else says sauce in their answer, right? And I know Colin is showing me what he understands, right? So make sure you can put your own flair on this, right? Just know, right, that I want to see that you understand. I don't want to see you regurgitating Google, right? Because that is not helpful for your understanding. You only have to turn in 3. Yes, Isaiah, right? So you could turn in 3 of the ones that we talked about right now. You could turn in the first 3, whatever you want to do. If you want to turn in all of them, that's fine. I am not going to be grading this based on what you put. I'm grading this based on, did you turn in 3 questions? And if I see a really glaring error, like you say DNA is a sugar, I might jump in, whoa, it's not, right? Otherwise, I'm looking for, did you try? The module exam is your next assignment after this, right? If you did all of these questions, or you at least went through them and made sure you understood each one, you're going to do super well. Okay, so if you don't have another question, let me get this straight, Bonnie says. Okay. I'll wait. Is this the DBA? It is. This was the DBA, Bonnie. It was a group DBA instead of each one of you having to call me, because that takes 7 million years and this takes 39 minutes. Well, you're getting credit for being here in the session and participating, which I'll go through who I got participating. You're turning in just 3 of the questions as evidence, yes, that you were paying attention. People that I had speak today were Colin, Joelle, Michael, Georgia, Elizabeth, Bonnie, Lane, Isaiah, and Briley. Did I miss anyone from that? Good question, Joelle. 3 of the questions from here. You don't have to create your own, just 3 of the ones here. So for example, you could do the first 3. You could do 3 of the ones that we talked about. You could do other random ones. You could put your finger on the screen and pick one, right? I don't really care which ones you pick. Personally, I would do the 3 that were harder for me, right? So that you can practice answering them. Last call for missing anyone that talked? I don't think so. You're going to be getting full credit for your DBA. If you did not speak, right, or you were not here, you're going to have some pretty good answers to your questions. That's what I'm going to be grading you on. If you were here, you're basically getting a free pass at the DBA, right? Of course, if you do have a question, include it, right? So Joelle, if you have your own question, you can put it in this too. I'll answer it. All right, anybody else confused on what you're doing? You're picking just 3 questions to turn in. You are getting a grade based on whether you participated today and you submit 3 questions. Okay. If you have additional questions, please email me, right? And otherwise, I will see you guys next Tuesday at 2:30.